course title is Microbial Technology and the lecture title is Ames test. Topic discussed in previous lecture. Ever since genetically modified microorganisms. Ever since genetic engineering was discovered in 1970, the world has fully been under the era of the gene revolution, and the genetically modified microorganisms are widely exploited in so many domains, from agriculture, medicine, industries to the environment. Their products are the great importance of the man and its environment. However, their undesirable and ethical implications still remain a major worry of many minds, whether they should be accepted or not. The acceptance of the genetic engineering or its products is subjective to an individual's desire and the priority after making a cost benefits analysis. Precautionary measures such as risk assessment, monitoring and the management have been put in place by policymakers to ensure the safety of genetically modified microorganisms and their product, which you already dealt. In this topic, we covered introduction, Ames test, its history, principle of Ames test, general methodology of Ames test and carcinogenesis, importance, limitation, fluctuation test, summary, conclusion, etc. Introduction. Mutations and any sudden permanent change in the sequence of data. DNA. The DNA sequence of a gene can be altered in a number of ways. Gene mutation have varying effect on health depending on where they occur and whether they alter the function of essential protein. Broadly, mutation divide into two types. First, gene mutation and second is chromosomal mutation. Gene mutation and chromosomal mutation is divided on the basis of their specific classes. Gene mutation, a change in a gene at a single nucleotide pair, and the chromosomal mutation, a change in a gene due to alteration of chromosome structure of number. A gene mutation is also point mutation, which is due to the substitution, insertion, deletion, inversion, etc. And the chromosomal mutation is two types: chromosomal mutation on the basis of structural changes and the number of chromosomal changes. On the basis of structural changes, it is deletion, inversion, translocation, duplication, and the chromosomal number change basis it is aneuploidy and polyploidy. And the mutation on the basis of origin, spontaneous mutation and induced mutation, which is occur spontaneously, it is called spontaneous mutation, non-disjunction during meiosis and DNA replication. Whereas induced mutation is caused by the mutagens, which is either physical or chemical, such as rays and different types of chemicals, that is organism exposed to the mutagens. Then occur induced mutation. Classification of different kind of mutation. First is depending upon the cell types is somatic mutation and second one is germinal mutation. Mutation that occur in the somatic tissue of the body is called somatic mutation. Mutation are not transmitted to progeny. The extent of the phenotypically effect depends upon whether the mutation is dominant or recessive. Next is germinal mutation, mutation that are in the germ tissue of the body. Mutation may be transmitted to progeny. Dominant mutation are seen in first generation after the mutation occur. Mode of origin is the base of second type of mutation classification. On the basis of mode of origin, it is two types: spontaneous mutation and induced mutation, which we already dealt. Induced mutation is due to the mutagens, and mutagens may be chemical agents or physical agents. If it is chemical agent, it is divided into two categories: ionizing radiation and non-ionizing radiation. Under ionizing radiation, it is particulated and non-particulated. Under particulated it is alpha ray and non-particulated it is X-ray etc. Whereas in non-ionizing radiation it is UV rays. In chemical mutagens it is alkylating agent, for example EMS, MMS etc. Another one is base analogues, for example 5-bromo uracil etc. Now, on the basis of direction of mutation, it is divided into two parts: forward mutation, reverse mutation. Reverse mutation is also called back mutation. Forward mutation in all organism, when mutation create a change from wild type to abnormal type, then the type of mutation are known as forward mutation. Most mutation are forward mutation. Reverse mutation or back mutation are often corrected by the error correcting mechanism, so that an abnormal phenotype change into wild type phenotype. On the basis of size and quality, it is further divided in point mutation and multiple mutation. Under point mutation, when heritable alteration occurs in a very small segment of DNA molecules, that is a single nucleotide or nucleotide pair, then these types of mutation are called point mutation. Next, the point mutation may occur due to following types of subnucleotide change in the DNA or RNA, that is deletion, insertion, inversion or substitution. Deletion, the point mutation which is caused due to the loss or deletion of the some portion of single nucleotide pair in a triplet codon or cistron of or gene is called deletion mutation. Insertional mutation, the point mutation which occur due to the addition of one or more extra nucleotide to a gene or cistron are called insertional mutation. Next one is inversion, when some gene sequence inverted or inserted back into the original sequence, it is called inversion. Substitution, a point mutation in which a nucleotide of a triplet is replaced by the another nucleotide is called substitution mutation. Now, under this point mutation, some mutation are also known as, first one is silence mutation, second one is missense mutation, third one is nonsense mutation and fourth one is frame shift mutation. It is a non-expressive and insilent mutation, a new codon codes for the same amino acid as the wild type one. In missense mutation, a codon originated from a nucleotide chain, then while code for different amino acid, it can lead to alteration or loss of function in protein. In nonsense mutation, a stop codon is adding to premature protein, it stops protein synthesis, because a stop codon ends synthesis of proteins and result in a premature protein or truncated protein. Frame shift mutation, the mutations which arise from the insertion or deletion of the individual nucleotides and cause the rest of the message downstream of the mutation to be read out of phase are called frame shift mutation. Next, mutation classification on the basis of phenotype effect. It is three type: morphological mutation, lethal mutation and conditional mutation. Morphological mutations are mutations that affect the outwardly visible properties of an organism. For example, curly ears in the cat. Next, lethal mutation are mutations that affect the viability of the organism. Then man versus Max cat. The conditional mutation are mutations in which the mutant alleles cause the mutant. According to the phenotype effect, following kinds of mutation may occur. First, dominant mutation, second is recessive mutation and third one is now dominant mutation. The mutation which have dominant phenotype expression are called dominant mutation. Recessive, most type of mutation are recessive in nature and so they are not expressed phenotypically immediately. In the isoalleles, some mutation alter the phenotype of an organism so slightly that they can be detected only by a special technique. Chromosomal mutation and types. Chromosomal mutation on the basis of a structural change. First is change in number of genes. either deletion which involves loss of a broken part of chromosome. In addition, duplication which involves addition of a part of chromosome. Changes in gene arrangement. First, rotation of a group of genes 180 degree within one chromosome. Inversion, in which broken segment reattached to original chromosome in reverse order. Exchange of part between chromosomes of different pairs. Translocation, in which the broken segment becomes attached to a non-homologous chromosome, resulting in new linkage relation. Second, in change in the number of chromosome. On the basis of change in number of chromosome, it is divided into two, euploidy and polyploidy. Euploidy, it involves the loss or gain of whole chromosome set. The euploidy are those organisms which contain balanced set or sets of chromosome in many numbers. The number of chromosome in a basic set is called the monoploidy numbers, denoted by X. Those euploidy types whose number of sets is greater than 2 are called polyploidy. Thus, 1x is monoploidy, 2x is diploidy, and the polyploidy types are 3x triploidy, 4x tetraploidy, 5x pentaploidy, 6s hexaploidy, and so on. Next one is polyploidy. It is further divided into two on the basis of addition of one or more set of chromosome. Autopolyploidy and allopolyploidy. The autopolyploidy involves polyploidy in which the same basic sets of chromosome are multiplied. Or allopolyploidy, the polyploidy result due to the doubling of the chromosome in a F1 hybrid which is derived from two distinctly different species. The resultant species is called allopolyploidy. Now, after discussing the part of mutation, we understand the Ames test because we use this Ames test for the detection of mutation. The Ames test is a commonly used method that utilizes bacteria to test whether a particularly chemical can cause mutation in the DNA of the test organism. It is also called bacterial reverse mutation assay. It is biological assay that is formally used to assess the mutagenic potential of the chemical compounds. A positive result from the test will indicate that the chemical is mutagenic and therefore may act as a carcinogen, as cancer is often linked to mutation. The test also serves as a quick and convenient assay to estimate the carcinogenic potential of a compound because standard carcinogens assay on mice and rats are time consuming, taking 2-3 years of the complete and the expensive. Although it is also noted that false positive and false negative are known. The Ames testing is also used to test water extract from the plumbing products in contact with drinking water using Salmonella typhi histidine reverse system, which involves mutation of the histidine locus in the genome of the several strains. This test is conducted in compliance of the Organization for the Economic Cooperation and Development 471 guideline for testing of chemicals bacterial reverse mutation test. Abbreviation assay are available for the certain situation. For example, the modified Ames oils test and mutagenic activity of the water extracted test AS/NZS 4020. History. The Ames test in the 1970s by Bruce Ames, professor of the biochemistry at UC Berkeley, as a fast and sensitive assay of the ability of a chemical compounds or mixing to introduce mutation in DNA. Because the assay does not use a live animal model, it is inexpensive, easy and fast. Bruce Ames published with his work in a series of papers including identifying environmental chemicals causing mutation in cancer in the Journal of Science, volume 204, 1979. Dr. Ames' work was critical in linking mutation in DNA to carcinogenic. His work identified many mutagens including pesticides such as DDT, the food additives AF2 no longer used, a flame retardants, tri-BS and the mutagenic compound in commercially hair dye. The Salmonella typhimurium bacteria used in the Ames test have mutation in gene related to histidine production. These strains are mutant that are auxotropic, meaning they are unable to produce histidine yet need it for their growth. In order to enable cell to grow on the histidine free media, the approaches access the examined substance potential to cause mutation that lead to a return to a prototrophic condition. Now, the principle of Ames test. The test is based on the principle of reverse mutation or back mutation, which we already dealt. So, the test is also known as bacterial reverse mutation assay, in which we require two main things. First one is the test organism. Ames test uses several strains of bacteria, Salmonella or E. coli that carry mutation. That is, a particular strain of Salmonella typhimurium carry mutation in gene that encodes histidines. So, it is in an auxotropic mutant which lost the ability to synthesize histidine and amino acid utilizing the integrated ingredients of culture media. Those strains are known as His and required histidine in growth media. Second one is culturing His. Salmonella is in a media containing certain chemicals causes mutation in histidine encoding gene such that they regain the ability to synthesize histidine, His plus. This is the reverse mutation. Such chemical responsible to revert the mutation is actually a mutagen. So, this Ames test is used to test mutagenic ability of varieties of chemicals. For routine mutagenic testing, the Salmonella tester strain recommended are TA97A, TA98, TA100, TA102 and TA1535. TA1535 is optional and TA1537 is replaced by TA97A. General methodology. In order to detect mutagens acting through distinct pathway, the tester strains such as TA1537 or TA1538 or strain TA1531 are specially designed to detect either point or frame shift mutation in the genes necessary to synthesize histidine. Certain chemicals are very selective, resulting in inversion in a limited number of strains. In addition, the test strains have mutations in the mechanism of excision repair to increase the test sensitivity and in the gene encoding lipopolysaccharide production, which makes the bacteria's cell wall more permeable. The metabolism of the larger organisms such as mammals have a capacity to change a substance that is thought to be not mutagenic into one that is, or vice versa. Enzymes, rat liver can therefore be added in an effort to mimic the metabolic process effect upon the chemical substance during Ames test, hence improving the efficacy of testing a chemical substance mutagenicity with respect to larger species. Rat liver extracted inoptionally added to mimic the effect of metabolism because many substances such as benzopyrene only causes mutagenicity when they are metabolized. A minimal histidine plate agar is used to spread bacteria. The minimal quantity of histidine present in the growth medium facilitate the initial bacterial growth and gives them the chance to undergo mutation. Only bacteria, those are capable to synthesize their own histidine, can survive once the supply of histidine is exhausted. Incubation period is 48 hours. The substance mutagenicity is directly correlated with the bacterial colonies number it produce. Examples of chemicals that give positive response to Ames test: first one is 2-aminofluorescence, second one is ethylene dibromide, third one is geranium saffrole, fourth one is saccharine, and fifth one is aflatoxin. This is the figure which is shown the general procedure of Ames test. Ames test and their carcinogenous mutagens identified via Ames test are possible carcinogens. Earlier study showed 90% of known carcinogens. Later study showed 50 to 70% carcinogens. The dose response curve obtained in Ames test is almost always linear, indicate that there is no threshold concentration of mutagenesis, that is indicate there may be no safe threshold level of the chemical mutagens or carcinogens. The curve of dose responses employing different chemical concentration is almost always linear, suggesting that there is not a threshold or level of mutagenesis. This is integrative finding from the Ames test, thus it implies that there could not be permissible limit of mutagenic chemicals or carcinogens similar to the radiation. Nonetheless, some have suggested that organism may be able to withstand low concentration of mutagens because of mechanism of defense such as DNA repair. As a result, there might be threshold of specific chemical mutagens. Ames test personally oppose the linear doses response. Extrapolation of the high dose utilized in animal carcinogenesis. Experiment to the lower dose of chemicals often found in human exposures, since the artificially induced mitogenic response and could lead to false positive results. Importance of the Ames test. Ames test is one of the tests which screen for the potential chemical carcinogens. Under Pesticide Act of USA, there are 8 tests and Ames test is one of them. Similarly, Toxic Substance Control Act in USA, there are 6 tests and the Ames test is also one of them. Limitation. Salmonella typhimurium is a bacterium and thus not a perfect model. Ames are added to the test. In order to evaluate the mutagenic capacity of the metabolites generated from a parent molecule, instead the heparin system, S9 fraction and the rat liver is used to mimic mammalian metabolic environments. However, there are metabolic differences between rats and human that may impact the mutagenicity of the compound under test. Human liver S9 fraction may therefore enhance the test. Its uses was previously restricted by the accessibility, whereas it has recently become commercial, feasible and thus may be more practicable. Now, flocculation test. It is a technique which is also used for the detection of mutation. Agar plate on the plate includes technique where used in the development of the Ames test are previously used. A flocculation method has been created since then as an alternative method to doing the Ames test. This method is conceptually similar to the agar based approach in that it involves adding bacteria to a reaction mixture containing a small quantity of histidine, allowing the bacteria to proliferate, undergo mutation and eventually being producing histidine on their own. A pH indicator is used to count the number of wells in microplate that have changed color, which is the result of the metabolic activity of the reproducing bacteria lowering pH. It allows researchers to measure the mutation frequency. To determine a substance genotoxicity, the procedure is similar to the conventional Ames test in that the sample is concentrated with the normal reverse mutation rate in nature. The flocculation test. The fluctuation test technique is used exclusively with liquid culture, and it is scored by calculating the proportion of the wells in 19 or 384 wells microplate that change from purple to yellow. Out of 96 wells, number of the wells that have changed color in the mutation frequency is in the 96 well plate technique used to calculation. In order to identify any significant variation between the average mutation rate and the one used for the test sample, the plates are incubated for a maximum of 5 days. During that time, the number of mutated colony, which is yellow, is counted daily and compared to the background rate of the reverse mutation using pre-established table of the significance. In terms of accuracy and sensitivity, the fluctuation technique is comparable to the conventional pour plate technique. However, it has several advantages over the latter, including the need for less testing material and simple colorimetric endpoint. However, moreover, determining the quantity of positive wells out of 96 or 98 feasible wells is much quicker than counting single colonies on a plate of agar. Numerous commercial kits are available for there. This majority of kit includes lyophilized bacteria and other consumable in ready to be used state, and multi-channel pipette can be used to conduct testing. The fluctuation technique's sensitivity is increased and its application to minimal environmental mutagens is expanded by enabling the assessment of the larger amount of aqueous sample up to 75% volume by volume. This is the figure which is show fluctuation test in bacterial reverse mutation in a liquid culture. Now, summary. The Ames test is also known as Salmonella typhimurium reverse mutation assay. Is a quick bacterium test that uses the endpoint of the mutagenicity in bacterium to identify carcinogens. Mammalian metabolism has a role in triggering pro-mutagens. There is a strong albeit incomplete link between the Ames test mutagenicity and animal carcinogenicity. The latter finds mutation is a bacterial strains gene which generate a histidine depending strains when the original strains needs histidine. One of the toxicological tests that is used the most commonly is the Ames test. The assay is employed to test nearly all novel chemicals and medicinal compounds utilized in business. This is the reference regarding Ames test. Topic to be covered in the next lecture. In next lecture we will going to study about the virus and its genetic system. In this topic we will study about the basic structure of virus, its genetics, studying virus that infected bacteria. Experiment with the viruses provide key evidences that genes are made of nucleic acids, and we also studied the genetic replication mechanism as well as transcription and translation process in the virus. Viruses have a unique genetic mechanism that help us understand in viral disease. We also studied that viruses are used as agents of gene transfer during gene therapy. Myself Dr. Sabiha Naz, Assistant Professor, Department of Biotechnology and Microbiology from Bhilai Mahila Mahavidyalaya. Thank you so much.